Hello everyone, welcome to our biology classes. Here is Radha, and in this session we're going to discuss about the topic Golgi operators. Okay, so basically, as we all know, Golgi operators or can be called as Golgi complex or Golgi bodies, right? So they are present in the all eukaryotic organisms like animals and plants. So what is Golgi operators or what are Golgi complex or Golgi body means? It is one of the membrane-bound cell organelle present in the cytoplasm of all eukaryotic cells, right? So in different eukaryotes, we can observe different number of Golgi operators. For example, in animals, we can see different number of Golgi operators. While in case of plants, we can observe some more different number of Golgi operators. Okay, so in our body, even having different kind of cells, right? So if we consider the animals as example, so animals are made up of different kind of cells, right? Like cardiac cells, bone cells, muscle cells, and all, right? So different type of or different kind of cells are having different number of Golgi operators. So number of Golgi operators present in the particular cell depends on the process or activity of the cell. Okay, now we will see the definition. So it is one of the membrane bound cell organelle, right? Membrane bound cell organelle present in cytoplasm of all eukaryotes. Okay, now in this session we are going to discuss the label structure of the Golgi apparatus and also the functions. Okay, now let us draw a simple diagram of Golgi apparatus or Golgi complex or Golgi body. All are same. So the Golgi apparatus commonly present near to the endoplasmic reticulum. So it is commonly observed like this. Right, a bulged portions are present towards the or at the end of the Golgi apparatus. So Golgi body is one of a cup-like structure. Okay, this is the basic structure of Golgi apparatus, right? Now we will see the labelings. So this is called as, so the region present at the inside, so the region present towards the two lateral stands, that is called as lumen. And here we can observe a bulged portion that is called cisternae. And also we can observe two phases in the Golgi apparatus. So this is the cis phase and this is the trans phase. Okay, so this is cis phase. And also we can observe some more vesicles. Here we can observe the vesicle. So this phase can be called cis phase. And this phase can be called trans phase. Okay, and also here we can also observe the vesicles, right? The presence of vesicles or lysosomes. So these are the incoming vesicles, and towards the face of cis we can observe the incoming vesicles. So these are the incoming vesicles, and these vesicles can be called as the vesicles present at the side of trans phase. Those vesicles can be called as outgoing vesicles. Okay, this is the basic structure of the Golgi apparatus. Okay, now we will see the functions. Okay, so here we can observe two phases. This is the cis phase and this is the trans phase. So the vesicles are also we can observe, right? So those vesicles are made up of carbohydrates, proteins, lipids, and other nutrients. Okay, those vesicles are comes from the one of the membrane bound cell organelle that is called endoplasmic reticulum. So endoplasmic reticulum modifies so that may synthesize the proteins and lipids by the help of ribosomes. So proteins are synthesized by the help of ribosomes in the form of rough endoplasmic reticulum, and lipids like phospholipids and cholesterol, those are synthesized by the help of smooth endoplasmic reticulum, and those lipids and proteins can synthesize and forms into a vesicle like structure. Those vesicles are comes to the end of the Golgi apparatus. So it acquires the or it accepts the vesicles comes from the endoplasmic reticulum. So later we will see the functions. So the vesicles accepted in the phase, which phase? Cis phase. Though the vesicles comes from the endoplasmic reticulum are accepted in the phase of cis. Those vesicles enter into the Golgi complex through the cis phase. Okay, the entered vesicles will later modify and formed into smaller vesicle like structures, and some are will forms into a mature vesicles, and some vesicles will move back to the endoplasmic reticulum. These are the major procedures which are seen in the Golgi apparatus. Okay, now we will see the functions. So what is the major function of Golgi apparatus means? So it packages the synthesized proteins and lipids. It has the major function is packaging of proteins and lipids. Okay. So the proteins and lipids are further moves into some more other destinations in the cell. Some proteins and lipids will move outside the cell, and some proteins and lipids will stay inside the cell because they have to sort out or they have to modify further. That's why they stay inside the cell. So the completely modified or mature proteins and lipids will move out from the cell by the help of plasma membrane or cell wall in the plant cells. Okay, so and one more function of that Golgi apparatus is it transfers the proteins and lipids to other destinations. Other destinations of cell. Okay, and also we can observe some other functions of the Golgi apparatus as the vesicle like structures are present in the Golgi apparatus, right? So these are the acquired or the vesicles comes from the endoplasmic reticulum. That means incoming vesicles. Those incoming vesicles are comes from the endoplasmic reticulum. Okay, these vesicles are bigger in size than compared to the vesicles formed in the Golgi apparatus. So the endoplasmic reticulum, the vesicles from the endoplasmic reticulum are bigger in size. Okay, those vesicles will enter into the lumen region. So those vesicles entered into the lumen region and they break down into smaller molecules. So these bigger vesicles will break down into smaller molecules. Those they may be proteins, carbohydrates, or any other lipids. Okay, and they get modified and sought out from the Golgi apparatus for the further modifications in the cell. Okay, why these bigger vesicles will get breakdown means due to the presence of enzymes. So in the lumen area we can see the enzymes. Due to the presence of enzymes, those enzymes will break down the incoming vesicles. Those vesicles come from the endoplasmic reticulum, and these vesicles later come out from the, so outgoing vesicles means these vesicles are the modified vesicles of the endoplasmic reticulum. So these are the breakdowned molecules, breakdowned by the help of Golgi apparatus, where we can observe the enzymes in the lumen region. Okay, so the outgoing vesicles may go to other destinations like out of the cell and other organelles or any another part of the cell. Okay, this is the function of the Golgi apparatus. So it is the outgoing, here we can observe the outgoing vesicles, and here we can observe the incoming vesicles. Incoming vesicles are from the endoplasmic reticulum and which are bigger in size, and outgoing vesicles are from the Golgi apparatus which are smaller in size. Those are modified in nature. Okay, and also we can see discovery. We will see who discovered the Golgi apparatus. So Golgi apparatus is one of the first organelle observed in the cell. So this Golgi apparatus was observed by a great physician, Italian physician, named as Camillo Golgi. In 1897. So Camillo Golgi is one of a Italian great physician who observed the Golgi apparatus. So Golgi apparatus by the process of staining, by the techniques or process of staining in the cell. Okay, so he just observed the Golgi apparatus and he just named that as a Golgi cell organelle in 1897. So after 1 year, the Camillo Golgi was published that observed cell organelle as Golgi complex. In 1898, it was published. As Golgi apparatus or Golgi complex. Okay, so this is the information about the Golgi apparatus, and some more information we need to know that. So in animal cells we can observe different number of Golgi apparatus in different number of type of cells, as same in plant cells, right? So Golgi apparatus are present morely in the active cells like muscle cells, cardiac cells and all. Okay, in case of animal cells we can observe, so this whole structure can be called as Golgi stack. Okay, so in an animal cell we can observe 10 to 20 animal Golgi stacks. So we can observe 10 to 20 Golgi stacks are present. In case of animal cells. So those 10 to 20 Golgi stacks are present in a network-like structure. Those 10 to 20 Golgi stacks are connected by a tubular-like structure. Those connection is between the cisternal region. So this is the information about the Golgi apparatus, and also in plants also we can observe different number of Golgi bodies. The main function is the packaging and transferring of the protein and lipid molecules. Those are comes from the endoplasmic reticulum. Okay, a cell function also depends upon the Golgi apparatus. So this is the information about Golgi apparatus. If any doubt, please ask in the comment box, and thank you so much.